All right, so let's continue on with some cell transports and look at a few other options that we have for getting stuff into the cell. So now we know that water, carbon dioxide, and oxygen all move through the cell membrane, water specifically through osmosis, the other one through just plain old diffusion. Um, how do we get larger things in? Like our carbohydrates and our lipids, our proteins, nucleic acids, those all have to be able to move in and out of the cell. So how are we going to get those around? They're much, much larger than water, and they're much larger than oxygen and carbon dioxide. So there's got to be another way to do this. The cell membrane is full of what we call signals, and they're kind of like doorbells, and there's tons of them. Now remember, at the molecular level, shape is extraordinarily important. If it's not the right shape, it's not going to be able to do what it's supposed to do. So here's an example of our cell membrane. And you can see down here at the bottom, here's our phospholipids. There's two layers of them, so it's our phospholipid bilayer. There's the water-loving part on the outside, the hydrophilic heads, and the water-hating parts on the midsides, the hydrophobic tails. And there's all kinds of proteins and other type of macromolecules embedded in, and just different combinations of them. And think of these as all specific little doorbells, all right? Each one's just different. Whenever a molecule floats around and it bumps into the cell membrane, it's going to press up against these little doorbells. And if one of them matches up with the shapes that are on the molecule, then it's essentially like ringing the doorbell. And the cell has the ability to detect these through that process and determine, well, which of these do we need, which of these do we not need, which of these do we want to bring in, and which of these do we not want to bring in. And so in that way, it kind of allows things to enter and turns away other things. And this is how your cell mediates, and it's selectively permeable, and allows some things to come in and allows some things to leave, and other things to not come in and other things to leave. You don't want a virus getting in here. It's going to bounce off. Hopefully it won't match up with anything. But you get some sugar. Yeah, you want the sugar molecules to be able to go inside. So they're going to bounce off, hit this, bounce on it a little bit. Hopefully they'll match up with something, and the cell will recognize it, know that it needs it, and pull it in. But there's a couple different ways we can get stuff through the cell membrane in this fashion, using these little signals. All right, so let's talk about passive and active transport, some of the easy ways to get some of this stuff in. All right, so the cell membrane helps get things in through basically two processes, passive and active. Passive does not require energy, so you can imagine that active transport does require energy. So one of the types of passive transport is just good old-fashioned diffusion. High concentration on the outside, they're pretty small, they can bump in. This would be like oxygen. This would also be like water, especially for osmosis would be water. But carbon dioxide and oxygen are to move through this way. So that's just a great way to do it, right? Get through good old-fashioned diffusion. But we also have another type of diffusion called facilitated diffusion. It's diffusion, move from high concentration to low, but rather than move straight through the cell membrane, these are having to go through facilitators. All right? Facilitators just help things happen. So there's a protein embedded all the way through the cell membrane, and these little molecules can't really go through here, but they can go through this protein. So this protein allows them to come through. Here's another one, just a different example of one. Another type of protein that allows and helps things to come through, kind of mediates and moderates what comes through. Yes, it's working by diffusion. No, it does not take energy, so it's passive, but it has to go through a protein rather than just straight through the phospholipid bilayer. So it is facilitated diffusion. A good way to think about this is, let's say you're walking your dog along a fence line. He's a little bitty dog, and he sees a squirrel, and he bolts and he gets off his leash, and he goes straight through the fence, right? Because he's small. That's kind of like diffusion. But you can't go through the fence. You have to go to a gate. That gate facilitates your entry into somebody's yard, apparently, to get your dog. That's what this is like. These are the gates. Now think about it. If the molecule was even larger, if you're like, instead of walking through, maybe you're driving your vehicle, well, they may have a larger gate to help you get in. And that's kind of what these facilitated diffusions do. They're very specific about what they let in, but they definitely help us get things in and out. So other than passive transport, we have active transport, and it does require energy. So why would it require energy? Well, if you're going with diffusion, you don't need energy. You'll simply move by spacing yourselves out. Active transport goes against diffusion. It doesn't go from high concentration down to low. It goes from a low concentration up to high. Example would be passive transport would be like floating down a river. All right, you're just sitting in a tube, floating down the river, having a drink or two on a lazy Sunday or Saturday afternoon. And then that's like passive transport. But let's say you get to a point and you realize, hey, my ice chest has detached from me and it's stuck in the bushes upstream. Well, you got to get back in the creek and you have to start walking upstream against the flow of water. And if you've ever done that before, you'll know you get tired very quickly. It takes a lot of energy to go up against the current to go get your belongings. That would be an example of active transport. Why would we want to do that? Why would your body need to pump all of one, instead of letting it diffuse out, pump all of one type of molecule to one side and all of a separate molecule to the opposite side? Well, there's this whole thing in your body called a nervous system, and it sends electrical impulses up and down it. How does the electric impulse, literally just electricity, stay on your nerve cell? Because our body is mostly water, and we know electricity diffuses in water. Why doesn't it just derail off of your nervous system and just go off into the surrounding water and diffuse? Why is it? What keeps it on the nerve cell? And there's this great little active transport pump called a sodium-potassium pump, right? Any of you ever get a Charlie horse before, or sometimes they're called a cramp in your leg when you're moving around and doing a bunch of stuff? So what happens is the sodium-potassium pump takes sodium ions, which are positively charged, and pumps them to one side, keeps all of them over there. It takes potassium ions, which are negatively charged, and pumps them to the opposite side of the cell membrane and keeps them all over there. So the membrane is in between a strong negative and a strong positive charge, and it gives the membrane, the phospholipid membrane of the nerve cell, a charge. So when the electric impulse comes down it, it stays on the membrane because the membrane's charged. When you get a Charlie horse, what's happening is you've run out of either sodium or you've run out of potassium. You don't have them enough to be able to maintain the charge on the neuron, so when the electrical impulse gets to it, it just dissipates into the muscles that are all around it and causes them to contract, thus causing the cramp. Because what's the number one thing they're going to tell you when you get a cramp? They're going to tell you to go eat a banana. Well, what are bananas really rich for? They're really well known for having this particular thing in it, and that's potassium. That's going to help balance out your sodium-potassium pump, and you're going to start feeling better. You're going to get a charge back on your membranes, and you're going to feel better. But that's all based on this active transport using energy, right? Energy in the form of ATP, 3-phosphates. When it attaches here, it donates a phosphate, or detaches a phosphate, donates the electricity there, and we get ADP and a phosphate. And later on, I go back to the mitochondria, and that'll be put together to form ATP again, right? So those are some good examples of active and passive transport, all right? Here's another example of those facilitated diffusions. I actually gave you the name. This one is called a protein channel or a channel protein, and this is called a carrier protein. So yeah, it lets things go through, but it's much more selective about how they get in and out. But both of these are facilitated diffusion, right? This one's moving through, but it's not going through anywhere. It has to go through this gate. These have to go through this gate. So they doesn't cost any energy, but these are just facilitated diffusions of ions through the cell membrane. So what about really big molecules? Really big stuff, or large, even large groups of things. Larger molecules, they have to be brought into the cell. And there's a process called endocytosis, and then sometimes our machinery in our cells produces things that they want to get rid of, and our stomach cells produce stomach enzymes, and they don't want to keep that in the cell. They need to be dumped out into your stomach. So how do we get those out? Well, endo and exocytosis. So with endocytosis, simply what happens is a group or a large molecule simply presses against this cell membrane. The cell membrane starts to fold and wrap around it, and then brings it in. It's pretty neat. It's very simple. Reverse that process and you have exocytosis. Let's say we have enzymes that have been produced in the rough ER, and they've been packaged by the Golgi apparatus in this nice little lipid package, and they need to leave. So they'll start floating up, kind of like the wax that's in a lava lamp. They just go, they just float up, and they'll eventually merge with the outer part of the cell membrane. When they do, the packaging literally just becomes more cell membrane, and it eventually opens up and releases the enzymes out into the stomach or whatever products made back out into the body. Very simple process, but this is for large things, endocytosis or exocytosis. So large things or large groups of things. So that's pretty much all we have for cell transport. I hope this has been helpful, and I look forward to talking to you next time.